is yours.
Okay, great. Okay, hello everyone. So I thought that today I would just talk. I will give sort of a general overview of what my lab does, because I think it may be more useful than going, very much in depth, talking in depth about any specific project, and also because my lab does a number of different projects that asks somewhat different questions. But in general, the research in my lab is on insects and insect neuroscience. We are specifically interested in the sense of smell, so how insects smell different things. And also we work a little bit on vision. And so this slide just summarizes, that we work on Drosophila olfaction down here. We work on mosquito, so malaria mosquito specifically, olfaction and vision. We also started doing some work on mosquito sensory ecology, so we basically do some field work to this end. We also work on olfaction in black soldier flies, so that these flies, the big guys. And we also work on olfactory neurobiology and ecology of bumblebees. But the first thing that I will briefly mention is this genetic tool development for insect neuroscience. So essentially, we all know that Drosophila is a genetic model organism, and so over the years, maybe more than 50 years now, people have been developing genetic tools for flies, and that the reason we use flies for a lot of different questions is because not because perhaps flies is ecologically the best organism, but just because we have these genetic tools. And so what I myself do, I develop some of the new genetic tools that we use in Drosophila, and we develop them in Drosophila. But then later, we, for example, introduce them in mosquitoes, and other people introduce them into other organisms as well. So, by the way, if somebody is interested in any of these aspects more than others, I'm happy to just skip and just go straight to something else, because like I said, I have a lot of points here, and I don't want to completely zap people with all this stuff. So if somebody has any preferences, then just let me know, maybe. So in the meantime, I can start with the genetic tools and flies. So I don't know if anybody in the audience works on Drosophila. Lena, no?
No, I think it's the most, it's our students, and they didn't work because for the pandemic, for the war, we don't work with Drosophila.
For sure. Okay, so in Drosophila, basically, we have these genetic tricks, these genetic tools that are called binary expression systems. And the idea here is that we take genes from another organism, which is evolutionarily very far away from the fly, such as yeast or fungus or bacteria. So we take the transcription factors from these organisms and we introduce them in the fly. And we hope that this transcription factor, when it's expressed, when it's turned into a protein in the fly, that it will not do anything, right? So we hope that the transcription factor will not have any targets in the fly per se. Now you may want to know, why would we do it if it has no targets? What's the point, right? So what we can also do, we can then, for example, if we take transcription factor from yeast that is called GAL4, we can then also, so yeast has a DNA sequence that this GAL4 binds to. And that's how it works in the yeast. So then we can also take this DNA sequence out of yeast genome that is called UAS and introduce it into the fly as well. And the idea here will be that if our fly has both GAL4 and UAS, GAL4, when it's transcribed and translated, it will only bind to this UAS sequence and to nothing else. And equally, we hope that none of the fly's endogenous promoters, none of the fly's endogenous transcription factors will bind to this UAS sequence that we introduced from yeast. Okay, and so that way we have these two main components, a transcription factor from elsewhere, for example, yeast, and the DNA sequence, the binding sequence for this transcription factor that also comes from yeast, or the same organism as the transactivator came from. And I will show you in a moment how this works. So what we also normally have is the repressor of this transactivator. So in the case of yeast, it's this galater repressor. And then again, in the case of yeast, the galater maybe we have a temperature sensitive version of it. So it doesn't work if we change temperature. It only works in cold temperature. So the same idea was applied to the transactivator and this transactivator binding sequence from E. coli, but in this case, this system is called Lex A system. But in this case, in the E. coli case, there is no repressor, so we cannot repress. We can only express a transgene, but we cannot stop it from working. And then the system I worked on is the third one, which is derived from the bread fungus. So here we have a transcriptional activator called QF, and the sequence it binds to is called QS, and there is a repressor. And this all can be regulated by quinic acid. So essentially the idea comes from, from this is the bread fungus. So this is a culture of it in the lab, and this is how it looks under the microscope. And this bread fungus can catabolize quinic acid. So it can use quinic acid as an alternative carbon source when its regular carbon sources, that is sugars, are not available. And it has a cluster of genes, which are shown here, which can catabolize quinic acid. And so the genes are regulated by this one transcription factor, and it has a repressor. And then transcription factor has the sequence that it can bind to, which is upstream of all these structural genes. So we're not worried about the structural genes. We're interested in these things which are highlighted in red here. So these transactivator and repressor and the transactivator binding sequence were cloned out of the Neurospora genome and introduced into Drosophila. And by the way, the Neurospora is a model organism itself. So people knew quite well what genes it has and what are the sequences of the genes and what the genes do. Okay, so the transactivator the sequence of it was cloned out of Neurospora, introduced into Drosophila. And in Drosophila, it is driven by a Drosophila specific promoter. So for example, I don't know, it doesn't matter what it is, but it's a Drosophila promoter. So to ensure that this transactivator will be expressed in some cells of Drosophila at some point in their life. Then we have another bunch of flies. Yeah, so this is one group of flies that sit in one tube. This is another group of flies sit in another tube. And they have this transactivator that, so the sequence that the transactivator can bind to upstream of our gene of interest that we want to express. And in the simplest case, this will be GFP, for example. Then we have a third group of flies that produce a repressor. And then we have quinic acid. So quinic acid is just, we can think of it as a drug. It's essentially powder that we could add to fly food, and flies can eat it. So what we have here is that if we have flies that have, for example, only this component number 2, the second stock of flies, they are genetically modified, right? But we would see no difference, no phenotype on them at all. Then if we have flies that have both component 1 and component 2, as shown here, this transactivator will be transcribed and translated. It will go and bind to this QS sequence, and this will switch on the expression of our GFP gene. So we will have GFP expressed in cells where promoter P1 is active. Now, if we add a third component here, if we add a repressor, essentially repressor deactivates transactivator and we have no expression again. And if we take flies of this specific genotype and feed them with quinic acid, the quinic acid acts on the repressor and it stops its action, and then we have expression of our GFP again. Okay, so this essentially allows us to switch GFP is just an example. Normally, it's not the GFP that we express. Normally, it's some toxin that can kill our cells, kill specific subset of cells. Or maybe it's an ion channel that allows us to artificially activate cells. Or maybe it's a receptor for something that these cells don't normally have, and so on. So GFP is just the simplest case, but normally we do some functional experiments. Okay, so like I said, this QF transactivator, it comes from bread fungus. And so when it was originally for the first time introduced into Drosophila, what we saw was that it had some toxicity problems. So we couldn't really get flies that would express it in all cells because these flies were dying. So this transactivator was doing something that we didn't understand. We didn't really understand. We didn't know why it was toxic to the flies. So what we have done, and that was some of my work, where we essentially modified this transactivator. So we removed the middle domain of it. And so normally this domain is a DNA binding domain, and this one is an activation domain. So this is the part of the protein to which repressor binds. And then there was this middle part, and we didn't know what this middle part does. So we removed the middle part. We also introduced a mutation on the C terminal here, which produced a somewhat weaker transactivator. But basically removing this middle part solved our toxicity problem. And we got a transactivator that essentially is not toxic to the flies, it doesn't kill them, but it still works fine, and it's still repressible. What we also produced is we produced so-called chimeric transactivators, whereby we fuse a part of GAL4, that's a transactivator from yeast, with a part of this, our transactivator from fungus. And these chimeric transactivators also work fine, and they allow for a broader range of genetic experiments that we can do in Drosophila now. And we have done the same, we created the same fusion with the transactivator that comes from E. coli bacteria and our fungus one. Okay, so another thing that we are working on in developing this Q system. So like I told you, we can feed flies with quinic acid, and that should switch the repressor off. However, so essentially what we are aiming for is that this is, for example, the brain of the fly, and you can see all these green cells. So there are cells that express GFP, okay? Then we add a repressor here. So we add an extra transgene that expresses QS repressor, and we see no green signal anymore. And that's because, well, repressor works. And the idea is that if we take these flies with this genotype, add quinic acid into their food, we should go back to this situation on the left here. However, that doesn't happen. What happens is that we go to this situation. So some cells are green, yeah, so they start expressing GFP. You can see these cells over here and these cells in this subesophageal zone of the fly brain. So this is the taste center of the fly brain. The expression of GFP, for example, in this region looks similar to this one, right? But the expression of GFP in most of the brain, for example, here and here, there's much less GFP than in the original situation. And so the reason that this is because quinic acid doesn't reach these cells for some reason. And the most sort of obvious reason why a drug may not reach cells in the brain is blood-brain barrier. So flies also have blood-brain barrier, which is essentially glia that is around the brain. And so we started working with a chemist in my university, Claire, and we had 2 master students who studied both chemistry and biology. So we started working on this hypothesis that the quinic acid doesn't reach some neurons in the fly brain because of the blood-brain barrier. And so one of the approaches, one of the first things that Claire suggested, and Claire's expertise is on drug delivery, by the way. So one of the approaches that Claire suggested is that we can not just add quinic acid into food, but we can embed this quinic acid into a liposome and feed flies with these liposomes mixed into food. And because liposomes are obviously lipophilic, this potentially could improve the fusion of the liposome with the membranes of cells and thereby deliver quinic acid, release quinic acid inside of the cells. So Harvey, our master student, he fed flies with these liposomes filled with quinic acid. And essentially he saw this situation, which is similar to what I showed you before, which is to say liposomes didn't help. Now, after that, Claire had another idea, which was that, you know, here we have the structure of quinic acid and here we have a structure of glucose, and they're similar. And so Claire's idea was that maybe the quinic acid can interact with glucose receptors that we know neurons and glia perhaps also have. And so we thought, okay, let's test this hypothesis by overexpressing the glucose transporter, GLUT1. So it's fly's native transporter. Fly has it, but genetically we can increase production of GLUT1 in glia or in neurons or any other subset of cells that we want. So first we said, okay, let's try to overexpress GLUT1 in glia, because the idea being, if glia is the problem and glia is what stops quinic acid, maybe by expressing more GLUT1 receptor on the membranes of the glial cells, we can take in more quinic acid into the glia, and thus hopefully it will pass the blood-brain barrier easier. This did not happen. So again, when we expressed GLUT1 in the glia, we saw exactly the same, you know, pattern as I have shown you previously. So there was basically no difference. And then Harris thought, okay, let's just express GLUT1 in neurons. I mean, why not? Because we can. And when Harvard did that, we saw that actually the expression of GFP is much higher now throughout the entire brain. And so that was very interesting. And it kind of indicated to us that blood-brain barrier is not a problem in this case. But what is the problem is that neurons themselves don't take up quinic acid. So it is available, it is around them, but they don't take it up. And we don't know why. But some neurons do, yeah. So these taste neurons which project here, they do take it up. So essentially now what we are trying to do, and this is what Charlotte is working on, is that we are trying to chemically modify quinic acid such that it binds to transporters or receptors which are naturally highly expressed in the fly brain. And other people have done single cell sequencing of fly brains, and so we know what transporters and receptors are highly expressed. So by looking at this data, I'm not showing it here, but there is a website, there is a database that can tell us what specific receptors and transporters are highly expressed in different subsets of cells in the fly brain. And so now Charlotte is working on essentially creating molecules which are a fusion of our quinic acid to this transporter or receptor that to the chemical that is binding to a transporter or receptor that is highly expressed in the fly brain. Okay, so while we were doing all this work on the development of this Q system, and this has been going on for about 8 years now, other people who work on other model organisms, they told us that our Q system also works well in other model organisms, such as zebrafish or C. elegans, HeLa cells, with the human cells, or Arabidopsis. And at that point, we became to be interested in mosquitoes, and specifically in the olfactory system of mosquitoes. And we thought, okay, let's try to introduce our Q system with the mosquitoes and see if it works as well, because there is a high chance, because it works so well in all these other organisms. So we produced first transgenic Anopheles gambiae mosquitoes. These are African malaria vector, with specifically targeted olfactory neurons. So you can see these green cells in this mosquito head here. So they are the olfactory cells in the mosquito brain that in the mosquito antennae that express GFP. And then the same approach was later used by other colleagues who targeted yellow fever and Zika and dengue vector, Aedes aegypti, and basically the approach was successful there as well. So Yeah, so now I'm going to the second sort of bunch of projects that my lab is doing, which is our mosquito work. So you might wonder why we, want to work on these mosquitoes in the first place, and why do we want to work on the olfactory system specifically? So the reason to work on mosquitoes is that they transmit human diseases. And malaria is one of the most deadly vector-transmitted diseases that we have. And we also know so we know that only female mosquitoes bite humans. And we know that the female mosquitoes use their sense of smell to detect CO2 that we emit, and also our body odor, to find us. Normally other cues are also involved. So for example, our body heat is also a cue that mosquitoes are seeking. And usually several of these cues need to be present at once for a mosquito to, decide that, okay, this object is probably a human. But their sense of smell is really important for host seeking. And that's why it's important for us to understand what they smell and how they smell. So another aspect here is that not much effort is going on now in the world to understand the neurobiology of the mosquito larvae. However, obviously, if the aim is to reduce mosquito biting and to reduce disease transmission, this reduction may be done at the adult stage, or interventions may be done at the adult stage, but also they may be done at the larval stage, because if you somehow reduce the number of larvae, or you do something with the larvae, this obviously reduces the number of adults that come out of this larvae later on. So the work that we started to do was thought, okay, first we just want to genetically label some of the olfactory neurons in mosquito. And as I said, we decided to use this Q system that comes from bread fungus for this purpose. And so essentially we created two transgenic lines of mosquitoes, which are shown here. We crossed them together. And here we are looking at the head of a male mosquito on the left and the female mosquito on the right. And this insert here, I hope you can see it, it shows, so all these green circles are individual cell bodies of olfactory neurons. And so we can see this in the live mosquitoes. There is no staining, there is no dissection or anything. We just literally put mosquito under the microscope with fluorescence and look at it. So what we could see is that our olfactory neurons are present in the mosquito antennae, maxillary pulps, and proboscis. And the number of cells that are present are strongly sexually dimorphic. So we have many more olfactory cells in the female antennae compared to male. We have many more cells in the female maxillary pulp. This is this orange number compared to male. And we also have these olfactory cells in the proboscis, which is the mouth part, but the number of cells in the proboscis are not sexually dimorphic. And this was perhaps a bit strange because, like I said, only female mosquitoes bite, and they bite with this proboscis. And so we thought that, okay, if there are sexual dimorphisms, they will be very pronounced in the proboscis, whereas in fact, that's not what we see, at least when it comes to the olfactory receptor neurons. Obviously, this proboscis also has the gustatory receptor neurons and lots of other, sensory neurons in it as well. So next, if we look at where these neurons, go in the brain. So this is a mosquito brain, and the green signal you see are the places which are targeted by the axons of these olfactory receptor neurons. So what we identified is that olfactory receptor neurons from the antennae and maxillary pulps, they project to this part of the brain, which is called antenna lobe. But the neurons from the proboscis project to a different part of the brain, which is called subesophageal zone. And this was again unexpected, because this is not the situation that we have in Drosophila. So in Drosophila, there are no olfactory innervation in this part of the brain. So in class, only antenna lobe processes olfactory information. And I always refer to Drosophila because Drosophila olfactory system is very well studied. And that's something we know well about. And we assume that, Drosophila olfactory system is a model for all insects, but in fact, what we are starting to see here, that even in mosquitoes, which are also diptera, so they're closely related to Drosophila, the situation is already quite different. Okay, so what we also saw here is that the olfactory anatomy is quite sexually dimorphic at the level of the antennal lobe as well. So here we're looking at the antennal lobe of female mosquitoes, and here on the bottom male mosquitoes, and these images are taken with the same magnification. So you can see that antennal lobe of female mosquitoes is about twice the size of that of the male. However, we don't see much dimorphism in this brain region where the proboscis neurons project to. So we don't see much differences in the subesophageal zone in terms of the olfactory innervations, which are shown here. And so this corresponds, this differences in the brain anatomy correspond well to the number of cells that we can count in the antennae and maxillary pulps, because we have more olfactory cells in females, and we also have larger antennal lobes. And equally, we have equal number of cells in the proboscis of males and females, and then we have very similar innervation patterns in the brain as well. Okay, so the summary of all what I just said is that we produced these transgenic malaria mosquitoes where we genetically targeted the olfactory neurons for the first time. And we expressed GFP in these neurons, and we count cells and look at the brains, and we see that there are strong sexual dimorphisms. Okay, so since we have these transgenic mosquitoes, we not only have transgenic adults, but we also have transgenic larvae. And like I said, nobody really much studies these larvae. And so Katie, who was a master's student in my lab, she decided to look at the olfactory system of the larvae. So this picture just shows larvae. They have 4 instars, so they have 4 developmental stages. These are the smallest ones. These are first instars, and then the larva grows, and becomes a fourth instar, and then it pupates. Larvae are aquatic, so they're in water. Pupae are also aquatic, they're also in water. And then adults are obviously terrestrial, so they're in the air. So when Katie looked at first instar larvae over here, and fourth instar larvae over here. In green here, you see these olfactory receptor neurons. So adult, as you saw, has hundreds of them. The larva only has 10 in its antennae. And the number of these cells in the antennae is not very different. So whether you look at, on the first instar or fourth instar is about 10 cells. But what changes is where the cell bodies of these cells are. So in the little larva early on, the cell bodies are actually inside the larval head. So the larval head the larval head is this. And then in these squares here are larval antennae, but the cell bodies are actually not in the antennae, they're inside the head at the base of the antennae here. And so gradually as the larva grows, these cell bodies move, and then by the time it reaches its fourth instar, the cell bodies are now here, actually inside the antenna. So the antenna is this. The head also becomes bigger, the antenna becomes bigger. But this entire time, you can see the dendrites of these cells innervate the very tip of the antenna, which is where the chemicals can enter and bind to the receptors on the dendrites of these olfactory neurons. So we also try to figure out where in the brain the axons of these neurons go. So essentially, we want to answer a question similar to what we answered already in the adults. So here you can see a 3D reconstruction of this larval head. And this white cloudy thing over here is the brain of the larvae. And in green, you can see these lines going towards the brain. These are the axons as we try to trace them. So this is something that we are still working on, because it was quite difficult to figure out where exactly they go. We use different imaging methods, but larval cuticle is quite thick and it has pigment. And so when we try to use optical imaging, it's actually quite difficult to image a significant distance inside larval head. But what we can see at the moment is that projections from the antennae and from the maxillary pulps, they kind of go in parallel. And so it suggests that perhaps the larval brain also has 2 olfactory centers, similar to what we see in the adults. So the next question that we are trying to answer in this larvae is not only where these cells are or how many cells are there, but we want to know what chemicals these cells respond to. And for this purpose, we're using live calcium imaging. So what happens here is that instead of expressing GFP in these cells, we express modified version of GFP that can bind calcium. So if there are more calcium in the cell, the fluorescent signal that is produced will be brighter. Now, when there could be more calcium in the cell? More calcium maybe when the cell is depolarized, and the cell is depolarized when it's activated perhaps by a chemical ligand. So essentially what it means is that when there is a chemical that the cell, a particular neuron can detect, it will glow brighter. And this is exactly what we see here. So here we're looking at the antenna of a mosquito larva, and you can see some cells start glowing brighter. That is because this mosquito larva is perfused with a chemical. It's sitting in a plate like this, it's glued to a dish, and we perfuse it with a chemical. And this chemical is detected by the neurons, by these specific neurons that start glowing bright. So this live imaging, this is our assay that allows us to see which cells respond to which chemicals. And this depends not only on the chemical identity of our stimulus, but also on its concentration. Because when we work with olfactory systems, concentrations are super important. So what the idea is that what we want to do, we want to be able to say, okay, these cells respond to these chemicals, but we also want to know what is the behavioral response of the larva to these same chemicals. Because we want to try to establish this connection between the sensory input and the behavioral output. And because these larvae have so few olfactory receptor neurons, we hope that it will be easier for us to establish this connection. So the problem here was that we didn't have a behavioral assay that would allow us to record responses to very precisely defined concentrations of chemical stimuli. So the assays that existed, they were like so. So one of them, essentially, they were, what people did, they were making two pieces of gel, like jelly. And one of them would be just clear jelly made from agarose in water, and another will also contain some chemical. And then these blobs of gel would be put in a dish. And, the chemical will start diffusing out of one of these blobs, and then we couldn't observe if larvae go towards it or away from it, and count how many larvae are, what distance away from this smelly jelly thing. What it doesn't tell us, so we know how much chemical we put into the gel, but we don't know how much chemical leaked out into the water. And so this assay doesn't allow us to do any sort of concentration dependent conclusions. Another assay that existed was a similar one where there's the square plate, and essentially people just pipette chemical into the corner, and the chemical will start obviously dissolve in water and diffuse. And then we can put larvae here and also see where larvae aggregate. But again, this is in no way telling us what concentration of this chemical larvae like or don't like, because we don't know what it is. And so Orshi and Matthew in my lab, they developed an assay that allows us to determine the behavior of the larvae in response to very clearly defined concentration of a chemical stimulus. So here the assay is actually very simple. We just have a 6-well plate, which is schematically shown here. In each of these wells, we have one larva. We place one larva. 3 of the wells have clear DI water, these ones, and 3 of the wells will have chemical dissolved in DI water at a known concentration. And obviously, we randomly assign which chemicals have clear water and which wells have clear water and which wells have a chemical. And then we can track these larvae for a defined period of time. We did 10 minutes. So we track larvae like so, and the way we interpreted these results is that if larva moves fast or obviously covers longer distance in 10 minutes, we interpret this as behavioral repellence. And if the larva moves slow or slower than the larva in water, we interpret this as behavioral attraction. And so this may be not a very sort of intuitively clear interpretation, but it has been substantiated by previous works in the literature by other groups. And so we sort of assume that we believe these people, we assume that what they tell us is correct. So, so what we saw, first we were trying to identify some attractive stimuli. And we used food which is infused with, a water which is infused with larval food. There will be no food particles, we filtered them out, but chemicals would be dissolved from the food into this water. And as you can see here, larvae moved less in this food-infused water compared to clear water. And so we interpreted this as attraction, and this is kind of what we would expect should happen. We saw a similar thing when we just dissolved glucose in water. We assumed larvae will like it because it's food. And we also infused some water with yeast, because larvae eat yeast particles. But again, there were no particles, there was just these chemicals which are dissolved in water. And in this case, larvae also looks like they like the stimulus. We also tested what we call home water, and this is just water from the trays in which larvae were bred, in which they grew. We took that water, it's of course not very clean, but if we filter it, it will still retain some, some chemical signature of their home environment, which supposedly they learned is safe and good. And smells of food as well, because we feed them in these trays where they grow. And so this looked like an attractive stimulus too. So essentially what it told us is that our assay can record or can report behavioral attraction. Next, we tested stimuli, pure chemical stimuli in quite high concentration. All of them were soluble in water. So we tested, you can see here, acetophenone, cyclohexanone, 1-butanol, DEET. DEET is an insect repellent, and isoamyl acetate. And at these given concentrations, all the stimuli were repellent, which we conclude from the fact that larvae move much more in the concentration of the stimulus compared to clear water. We also then did a series, we explored a series of concentrations of the same chemical, in this case isoamyl acetate. And we saw that when this chemical is at low concentration, such as 10⁻⁵ by volume, it's mildly attractive, then it becomes neutral, and then it becomes repellent as we increase concentration. And again, this is something that we would expect, because it's generally known that olfactory or chemical stimuli at high concentration are quite likely to be repellent. And at low concentration, if it's very low, we cannot smell them, obviously, but they may be attractive at low concentration, some chemicals. Okay, so next we wanted to address the following question. So we put this larvae in water and we see some behavior, and this is all grand. But we cannot really definitively say that this behavior that we see is mediated by olfactory neurons, because larvae are bathed in this water. They can drink it, it will activate their taste neurons as well. The chemicals may bind to whatever, some other receptors on their body that we maybe don't even know them. And so we wanted to ascertain that the behavior that we see is indeed due to olfactory neurons and not other receptor neurons. And one way we can do it is to silence the olfactory neurons and see how behavior changes. If behavior changed, it means olfactory neurons were important. If behavior didn't change, it means olfactory neurons were not important in this case, and some other neurons mediate this observed behavior. So in order to silence these neurons, we used another genetic trick that we know from Drosophila. So we can express in these cells this inward defined potassium channel. So what it is that it's an ion channel that pumps potassium out of a neuron. Potassium is positively charged. So if we push it out, it means neuron will become more negatively charged. And by doing so, we can push the membrane potential of the neuron so low that it will not be able to fire action potentials. So if a neuron cannot fire action potentials, it cannot tell the next neuron in line that something happens, right? So functionally, we disable this neuron, we shut it down. So So we produced these mosquitoes that express this KIR2.1 potassium channel in olfactory neurons. The neurons that you previously saw are labeled in green. And we picked the concentration of a chemical, acetophenone, here that is very repellent to wild-type larvae. So you can see wild-type larvae might move much more in this concentration of chemical compared to clean water. When we presented the same concentration of chemical to our larvae with silenced neurons, they didn't exhibit any difference between this chemical and water. And so this was quite exciting, and it told us two things. First is that it looks like larval olfactory neurons can indeed be silenced with this KIR2.1. This has never been done before, and obviously we didn't know it would work. And second, we learned that in case of acetophenone, in this specific case, the behavior we observe is indeed due to the action of these olfactory neurons and not other sensory neurons, which was also quite nice. We did a similar experiment where we tested responses to the insect repellent DEET. And in the case of DEET, what we saw is that the repellent response is reduced here, but it's not abolished. And so what it tells us is that DEET is detected by olfactory neurons, but not only. And there are other neurons, perhaps taste neurons, that can also respond to DEET. And this is something we would expect because we know from experiments on adult mosquitoes and also other insects that DEET, the action of DEET is very multimodal and binds to many sensory receptor cells. So finally, we wanted to know whether we can silence olfactory neurons in adult mosquitoes as well. And this was work of two other students in my lab. And so what Michelle and Natasha did was that they looked at how quickly mosquito escapes when it's approached by a filter tip with some chemical on it. And they saw that if we take wild-type mosquito, non-genetically modified mosquito, and presented with some 1-hexanal, it escapes very fast. And we presented with mineral oil, it doesn't really escape, it's just sitting there. And when they presented same chemical stimuli to mosquitoes with potentially silenced neurons, they indeed saw that these mosquitoes don't escape 1-hexanal anymore. So we interpreted it as a fact that the olfactory neurons don't signal that they are essentially silenced. And we saw the same result here with another chemical, with 1% benzaldehyde. So our conclusion from here is that we can silence olfactory neurons both in the larvae and in the adult now. Okay, so this is just a summary of what I have told you so far, is that we investigated the larval olfactory system, and there are very few neurons in the larval olfactory system compared to adult. They seem to be wired up and functional from the first larval instar onwards. We developed this new assay that can detect larval attraction and repellence to a range of chemical stimuli. We can now silence larval and adult olfactory neurons. And a little bit of new biology that we learned is that larval responses to acetophenone are mediated by olfactory neurons, but this is not the case with DEET. Okay, so that is our second project, this mosquito olfaction. Okay. Now another project that we're working on is that we're looking at the development of the visual system in mosquito larvae. So just very quickly, why this is interesting. So the mosquito larval eye starts out as this sort of structure called larval eye, and then you see here a second instar, and the first instar also, there's only this larval eye. So it's not like the usual insect compound eye, it's a different structure. But then what starts happening is that in the third and fourth instar, the actual compound eye, adult eye, starts developing in the larva. And so what, for example, here we see this is the fourth instar, the larva still has this larval eye, but it also has the developing adult eye as well. And so it creates this interesting situation where the larva essentially has two sets of eyes. One set are these larval eyes, and they express their own set of opsins, the visual, the light receptors. And then we have this adult eye, which is also there, and it's also supposedly wired up and functional and doing something. And then by the time we get to adult, so young adult, early in the adult stage, there is still a little bit of a leftover of this larval eye, and supposedly it disappears later on, although we're not entirely sure about this. And so there was a paper published that looked at the opsin expressions throughout the mosquito development. And we wanted to look at this in a bit more detail. So Matt, a PhD student in my lab, he started looking at the opsin expression through every day of the larval life. He harvested larvae and looked at how highly are different opsins expressed. And so this is still Matt's ongoing work. He sees that this, opsin 6 is expressed quite highly in the larvae, and then it really disappears in the adult, as we would predict from previous results, and other opsins become switched on. So because opsin 6 is so predominant in the larvae, we are now trying to develop a mutant mosquito where this opsin 6 will be mutated, and we want to see whether other opsins which are expressed at a lower level will still mediate any visual behaviors, or indeed opsin 6 is the only functionally important opsin in the larvae. So this just shows that, in adult males and females, there are opsins 1 and 3 that are also, that become highly expressed, and eventually they will be more strongly expressed than opsin 6 as we progress through the adult stage. So what we're also doing here, and this is a collaboration with Mike Kittelman from Oxford Brookes University, we decided to use micro-CT technique to take images of the larval and pupal heads to look at how the visual system develops. And so this is, again, a work in progress, but I just want to show you an example of such video. So we are looking, this is a Z-stack, and we're going through the Z-stack of a pupa, I believe, in this case. So we're going like this. And at some point, I hope we will see the brain and the visual system of these guys as well. So this technique is basically, it works as an X-ray in a way. So we are not really, well, the tissue is pre-stained with iodine, and then the picture is created by the diffraction of electrons. In this animal. So here we start, here we start seeing our big compound eyes. I hope you can see that, but this is in the pupa, right? And here is the brain as well. So here's the, we're going through the brain of this pupae as well. So we can take images like this from larvae of different stages and pupae in adults. And we can look at how different brain areas change in their size and volume, and also how the eye structure changes. So that's something that Matt is also working on now. Okay, so Lena, should I keep talking or should I stop? Because it's been a while now.
I think maybe some conclusions, Lena, and after that, maybe some of our participants will have some questions. I think maybe 5 minutes for some conclusions, or maybe something very interesting and some exciting factor. Or maybe
okay,
something.
So maybe maybe what I'll do, I'll go back to that slide that I showed you in the beginning, like this one, and I'll just say very briefly what we are doing with other insects. So with these black soldier flies, we started working on them because they are agriculturally or industrially important. And the reason they're industrially important is that, so the fly is big, the fly itself is like this, like half a finger, and the larvae are also very big. And these larvae essentially eat any organic material. And so they're used to process organic waste. And at the same time, the larvae themselves, when they're fully grown, they may be, harvested and used as a source of protein or lipids for various purposes, cosmetic industry or animal feed and so on. And so we started working with this company, which is in Edinburgh in the UK. And they told us that they would really be interested in having synthetic oviposition attractant. So they want to be able to collect eggs from larvae, many eggs in one place. Because normally what these larvae, these flies do, they naturally deposit eggs into various crevices in wool. But when they're in the lab, they're held in these cages and they put eggs into seams and all sort of small gaps and so on. It's very difficult to collect these eggs. And the company used an oviposition attractant, but the attractant was just flies, dead flies in water, brewed for a few days. So that was, you can imagine, not very nice. And most importantly, they couldn't give this attractant to a customer because, you know, customer doesn't want to deal with these dead flies in water. This is all very messy. And they wanted to have a chemically defined oviposition attractant. So just a mixture of chemicals that they can produce themselves and, you know, give to a customer as a prepared, as a ready formulation. And so what we did here, we analyzed the volatiles that are produced by these dead flies in water, and we identified many. And then a student in my lab, she created different mixtures of these compounds, and she managed to find a mixture of only 5 chemically defined compounds, which is very attractive to the flies. So this is just to say that we will manage to create a synthetic oviposition attractant, which is really interesting. And also in the process of this project, we found that actually nothing is known about the neuroscience of these flies. So there isn't even a paper that would like show you a picture of their brain or anything like that. And so we started working we started creating images of their brains. So we started immunostaining and imaging their brains. And we want to, well, since nothing is known, then anything that we describe or find will be informative, but we are still working on this. And so I have this bumblebee on the flower here. So with bumblebees, we also have a sort of chemical ecology project where so essentially in this area of England where we are, bumblebees are quite common because it's quite cold here. And so we have 7 or 8 bumblebee species that we can easily find in summer. These are 7 or 8 social, and there are some solitary ones as well. And so what we are doing is, the question we want to ask is how does the bee use the smell of the flower to decide that it wants to forage on this flower? So what we are doing is that we are going outside, we are seeing a bee, we are collecting a bee, and then we are also collecting volatiles, so the smell the volatiles from a flower that the bee was on. And we want to know whether there is anything in common between the smell composition of the flower that bee species forages on, and whether there are any differences between the smells that different species of bees prefer. And again, this is an ongoing project, and yeah, we don't have a definitive conclusion yet, but we think we do see some differences. So I would just like to, because I couldn't mention all people, I will just show you a picture of my group, because basically these are the guys who collected all the data, right? And I'm just talking here about it. Okay, thank you. I'm done.
Okay, thank you, Lena. It was very exciting, especially in terms that, it's a very interesting topic at all, the neuro investigation of insects. It's some very interesting for me, especially because I previously don't even think that it may be so interesting, and it's can
many
be
people don't know that insects have brains. So.
No, I understand that it's really possible to find these genes, to find these mechanisms, and to understand more better, because so many works at Drosophila translated to the humans, and maybe some of parts of these works can be translated to humans too. And I think that we have a question. Yes, Victor Alexeyevich.
Okay, should I
Yes,
stop sharing? Maybe, yeah, maybe I'll just stop sharing.
Yeah.
Hello,
Mhm.
hello,
Hello.
Lena. It was